When one thinks of DNA, one thinks of the standard right-handed, two-stranded double helix structure. This structure, known as bDNA, although it is the most common and well-known structure, is not the only form that DNA can take. There are two other well-known forms known as aDNA, which is like bDNA, but it's thicker and more tightly wound, and zDNA, which is a left-handed helix structure. However, all these structures are only two-stranded, and DNA can actually take on a variety of forms, including those that are four-stranded. One of these types of molecules are known as G-quadruplexes. These structures are actually thought to exist in all living cells because they have been found in both prokaryotes and eukaryotes. These structures are formed in high concentrations of guanine, which is one of the nitrogenous bases found in DNA. Similar structures can be formed in high concentrations of cytosine. However, these structures only form within acidic environments and are not very relevant to the biological functions of the cell. G-quadruplexes exhibit unique structural properties. They contain a lot more hydrogen bonds than the standard double helix DNA, so they are much more stable. They can withstand temperatures well over 60 degrees Celsius under physiological salt concentrations. Although relatively little is known about their biological function, they have been shown to interact with telomeres, which are DNA sequences at the end of chromosomes that regulate programmed cell death. Additionally, they're shown to be involved in transcription regulation because they've been shown to be capable of inhibiting the process. G-quadruplexes are of particular interest to cancer researchers because, as recently stated, they interact with telomeres, which regulate programmed cell death. Telomeres are actually closely related to cancer because in most of cancer cells, these structures are deformed, being much shorter than they're normally supposed to be. This causes instability in chromosomes and effectively immortalizes the cell, causing it to live forever and replicate out of control, as seen in tumor cells. This malfunction in the cell's chromosomes is facilitated by an enzyme called telomerase, which continues to add DNA segments to the end of telomeres, preventing the cell from dying. G-quadruplexes are of interest to cancer researchers because they are capable of inhibiting the enzyme by introducing chemicals that interact with G-quadruplexes, which then interact with telomeres. This can be used to slow down the division and the spread of cancer cells, allowing for specialized treatments that work against cancer more effectively. Among these anti-cancer chemicals is a series of chemicals called cationic porphyrins that have been identified as G-quadruplex interactive agents, otherwise known as QIAs, and they stabilize telomeric G-quadruplex DNA and thereby inhibit human telomerase. In an experiment, they were able to inhibit 50% of telomerase activity in a sample of human cells. Four-stranded DNA has shown to be incredibly important in the field of medicine, and its research may lead to groundbreaking treatments to one of the most widespread and devastating diseases to afflict humanity, and is a significant part of the field of biology.
闇を切り裂いて You are someone 俺の闇を切り裂いて誰も二人の安らぎ壊すことできはしないさ